Thank you very much, Beck. Good morning, everyone. So let's kick off today's program from the first talk. So thanks for joining the very beginning, the surveys online to have a understanding the audience in the rooms. I think we were, as Beck said, we prepared probably exactly what people need at this moment. So my first talk is going to be used as a very brief introductions for all of the beginners who have lack of experience in this field, just to explain what is actually neurofeedback, and then the examples of neurofeedback, and also the concepts and then the technical aspects, so that's help to further lead into the discussions later today. Again, as I said, so we were supposed to be co-leading this talk, and then the sessions with Professor Valentina Lorenzetti and myself, she's apologized and is in conference in Europe somewhere, so I'm just trying to cover the talk. As Beck said, my background is mainly physics and engineer, so I tried to use my way to look into the whole projects and the development of neurofeedback, and there'll be lots of focusing on the techniques. Okay, let me get the slide. Is this working this way? How do I go to the next slide? This one? Ah, yeah, that works. All right, cool. So a brief outlines of the talk today. So we're going to start with a brief of the neurofeedback journeys of our lab and our groups. And then we're going to touch base of some of the brief introductions of what is actually neurofeedback. And I'll give examples of some of our projects and on the different scanners, and how do we achieve, and there's some details of neurofeedback. And I'm going to review the whole structure of neurofeedbacks and what is the challenge of setting up a functional MRI-based neurofeedback study, and what's roughly the solutions it is. And then finally, we're going to quickly go through the plan because we're changing the plan of today's talk a little bit, so I'll explain to you what is actually going to happen this morning. So the journeys of us on neurofeedbacks is really starting around 10 years ago, so when myself and then Valis worked with Murat in the times in Monash, and then working in the brain and mental health society labs in Monash University. And then Vali get this unique opportunity to went to Rio in Brazil and work with the local team to upscale her neurofeedback. And then she set up a project with the local technical teams on the motion and emotion regulations on the 3D scanners over there. And then she brought this skill back and then replicated and set up the first MRI, functional MRI neurofeedback scenarios at Monash biomedical imaging facilities, which is a 3D Skyra Siemens scan. And I was involved with that project with her. And then a couple of years later, she moved to Australian Catholic University, ACUs, and to start this neuroscience of addiction and a mental health program. And then she's involved into this very successful setup of meditation, mindfulness neurofeedback projects, leading by Sampras, who is going to talk about more details, and with Andrew, Brad, and Rebecca from Uni of Melbourne. So we have this, I would say the third project is set up in the Uni Melbourne, the 7 Tesla scanners. And then about 2 years ago, we have Ethan and Amir's on board, and we're starting to build up our very recent functional MRI scenarios on the cravings and with addiction, etc. So this is our new projects, and we develop really quick, and actually it's just we finished the build up the whole scenario and get a couple of participants into the study. It's really new and fresh, and we have all of this experience here today to share with everyone. Okay, so that's just a little bit of background of where we're from, and then how we form into this neurofeedback. For lots of people, if you don't have any ideas of neurofeedback, so I'll just give you a very simple way to explain what it is. So we know this is, talking about function MRI. So this is a typical task-based function MRI. You got the participants lying down in the MRI scanner, and you give them a visual stimulus or some stimulus. So here, because we're working on addiction, so that's just a cannabis leaves, so there's. So we stimulate some maybe cravings, some activations. This is a basic like task-based function MRI. So it's the similar things as what you're doing with all of the task-based function MRI. And then you get the activations of the brains. And then the differences of neurofeedback is you trying to find a region of interest or somehow analyze the data and get those information and feedback into the participants while the participants was in the scanner. So the participants will actually see the activations or to put in particular part of the regions while he was reacting with the stimulus. So that's where the feedback is coming from. Okay, we're gonna come back to this loop later on. So if you really want to give a definition, so function MRI neurofeedback is a non-invasive neuromodulation technique whereby people receive real-time feedback on the brain function via a brain-computer interface, such as here is a thermometer, is a color bar, it's going up and down to indicating the activations going up or down. They can be different. We'll come into some other options later on. The brain and computer interface that which participants actually see, we also sometimes refer to the front end. Okay, so it's a similar like we asked those questions before. So neurofeedback is not only function MRI specific, it can be applied through any other neuroimaging techniques like EEG, MEG, the functional near-infrared spectroscopy. However, today we're just mainly focusing the function MRI-based one. Like this is an example of the EEG. For me, of the technical people speaking, so any kind of feedback, it's related with this, and this is a broader applications. And then this is actually a very interesting setup of the FMRI. When I was first time trial, probably that was nearly 8 or 9 years ago in human brain mappings, where you actually can get the activations of brain activities from the bilateral hemisphere, and then that sends you the feedback of the phone. And there's a gamified applications, and you can try to play with the games from what you think about it. So in other words, so you see the product is probably 10 years ago, so neurofeedback is not something really recent. It has been developed for quite a long history. All right, so this is the definitions again. So as we find, neurofeedback is considered as a neuromodulation technique. So you actually can thinking there's a lot of applications. So function MRI neurofeedback enable the real-time regulation of people's own brain function. So when the participants in there, they got the feelings of they can modulate and feel the way to changing or regulate brain. So they have a lot of implications in different psycho disorders or psychiatric disorders. So there's a famous review by 36 back in 2020. And then there's a few different cohort that we can use. So for example, Ethan got his paper reviewed on craving related on substance use disorders. And then the neurofeedback can be used in improve the motor functions in Parkinson's disease, and they can also regulate the mood functions in depressions, and also they can in ADHDs to trying to improve the response inhibition. So there's a lot of applications in different disorders. So what is actually can you feedback in here? So one of the straightaway idea you can think about is just the activation. So you draw a surface based on the prior or hypothesis-driven. You find the regions, find the MNI coordinates, draw a surface or whatever the way that you extract the activities from that region and get activated and then the signal changing, and then you can feedback to the participants. And of course, you can put into any other different type of the measurements. If you're familiar with function MRI, for example, connectivity, and you got two regions and find how synchronized with each other and generate function connectivity and then move up. And if you can exploring some other little more advanced idea, for example, you can from the whole brain levels and you can narrow down the activations based on some task on the day. And also you can try to do a whole brain levels and then machine learning, just trying to find the different patterns and use the distance from the hyperplanes to give the feedback. So that part is a bit advanced. I'm not going to touch base today, but that's just to show you the potentials. Okay, I prepared a few of the examples of what we had before. So this is one of the first experiment we set up in, I think it's in Monash. So with just to test the function, the neurofeedback function is working. So it's using a classic finger tapping paradigm. So and with a little bit difference of the front end design. So as people know, function MRI, so finger tapping is one of the most robust function MRI paradigms. So where people thinking of the tapping fingers, even thinking of tapping the fingers, you find the activation in the motor cortex very strong, especially the hand lobe. And then that's where the region it is. So, well, participants, okay, let me see whether I can start this video. Yep. So, as you can see, this is a pretty typical 3D Unity developed environment. So, at this moment, the participant couldn't control everything, but they were flying within around these castles and flying around, and then they will find a rock, and then this is time. The cue is finger tapping, and then the participants is trying to tap their finger or thinking of tap their fingers, and then the activation will be popped up at the hand lobe, which is the motor cortex, and then the activations will be detected and feedback into the Unity scenarios, and then the rock is actually will be lifted up. And then after the rock lift up, you go to, remember this, keep saying this is rest 1 and rest 2. So it's very critical, important that you have to want to build the baseline. So it's the rest. So it's the activation was flat. And then we have the triggers, and the participants can actually see the activations that has been implicated. So, yep, and then this is the first one, experiment. I see this person's successful move it. So this is just a part of the whole experiment. So which is the first time we try to set it up and make sure that everything, all of the different system is actually working. And then we go into the second experiment, which is focusing on the emotion regulation. So in this setup, we have 3 conditions. The first one we can see here is where so on the left side is where the neutral conditions, which is you're walking on the field is like just normal colors. And then the participants will jump into the another 2 conditions, which is the positive emotional and the regular emotion. There is an audio cue is actually comes to play the music, and the participant will know, okay, this is time to do the regulations of positive or negative emotions. And it depends on different levels of, depend on different conditions, the 2 regions, OFC or amygdalas, will be the key region of interest. And then the data, the activity will be samples and feedback into the actual field. And then the changing of that is actually the color. So the golden one is the positive emotions, where the more activations they got from OFC, the color will be changed. And then the negative one is showed by these purple colors. So let's look at these videos to give us rough ideas. So the participants, so this is a neutral conditions where you can't control of where you move, but you're just walking slowly into this field. I got the music, that's good. So it's actually a back play of some classic music and give people a very not quite comfortable feelings, and that's actually to trigger in, this is the down regulations. It's a negative emotion, so, and then the participants is trying to linking the emotion into the negative emotional scenarios, and then trying to, and then we're looking into the amygdalas and to get the activation. So the more activation they detected, and then the feedback as the color schemes of the field. And you see that colors become really purple. And then again, so then you were going back into the neutral condition, so we rebuild the baselines and wash out the previous effect. And then it comes the positive emotional state. And similarly, when it's detecting more activations than OFCs, and then that's the feedback will feedback into the persons as the colors of the field is changing. Yep, okay. And we also play with some other aspects of these scenarios. It's in the papers. If you're interested, you can come back. You can search and have a look. I'm not cover everything into these details. And then the third experience, third examples that we did is with Sampras to set up this really cool meditations neurofeedback project. So I think Sampras is going to give more details in the second talk. And yeah, until the last, the most important scenario setup is something based on the craving, as the group has a long term of research interested into the addiction research. So we will have our theories of which part of the brain is related with cravings and substance use. So we can predefine some of the brain regions, and we also have years to develop this robust functional paradigms to look into the brain activation related with cannabis-induced cues, cannabis-induced craving. And then all of this is pointing to some part of the brain regions. And then also with the recent reviews of how we can use neurofeedback to modulate the actually the other cognitive function. So we developed our scenarios, which is we're going to talk about more into detail today. So within this one, we can just roughly let people to know. So slightly different with the previous examples. The back end, we don't use the old front engines, and we use turbo VNC. Like lots of people put the interest in the survey of this at the very beginning. And also the front end, we didn't use Unity, so it's not fancy scenarios that participant can see, but be very targeting and specific on the cues. And then we use the thermometer with the color bars to indicating the activation of feedback going up and down. So the details will be later talked about by Amir to talk about the design and then the setup into details. Yep, but here I can show you the examples of how this is working. So before the video started, so you can see in the middle of the screen that's showing get ready to decrease the craving bar. So this is actually the STEAM PC, if you understand what does that mean. So that's the screen that the participants actually can see when he's lying down in the scanner. So this is what we call as the front end. So it's talking with the participant directly. On the left side, that's we usually call the back end. So that's where the TBV was running, the Turbo Brain Voyager is running, which is the one is that actually calculate the feedback in the real time, and also listening to all of the data coming from the consoles and do the process and push it back into the front end, so the front end can present the activation or the feedback. Yeah, I think the volume is a bit low, but there's actually, well, you can see if this is louder, you can see actually the scanner noise is there. So this is what's actually happened while the scanning, during the scanning. As you can see, with the participants, you see the cues of the cannabis pictures, and then the craving, which is the activation of his ACC is increased, and he's trying to use some strategy and to bring the activation down. The participant actually doesn't know exactly which part of the brain region. He's just trying different strategy and reduce the activation. Okay, just to sum up for all of the setups in here, so we got we have currently experience of 4 different projects setups. It's across the 3T and the 70 scanners, and with the different softwares of the front end or back end. And also, it's very important we have set up and we can adapt and to roll out. So that's in the last talk that Elena, who is going to deliver online as well, so unfortunately she's apologized, but that's an example that she's just applying current frameworks into her research topics on the depression. Okay, so that's just another brief views, and then now I'm just trying to go into the kind of the frameworks, the technical frameworks of how to set up this function MRI neurofeedback project. So I always start with this slide. It's called behind the scenes. As this is, I'll start a video. This is the one that we did at Monash Biomedical Imaging on this motion regulation scenario. Yes. As you can hear, the music is actually playing. It's indicating the cue of going the negative emotion, and then the participants is actually trying to go into the negative emotion state, and then the activation of the amygdala has been detected, and then the color has been changed. So as presented before. But here I want to highlight is the functional. The neurofeedback scenario is sometimes it's a bit different as the other function MRI or MRI study in general. So, which is you can set up everything and then just relax, and then the participants were there collecting the data. But you can see it's always quite intense in the console rooms to have everything set up and make sure all of the work is actually working. So there's just so many times having mistakes or errors, we have restarted. It's quite intense when it's during the data collecting phase. I just want to highlight one of those little things, if you see that. So this is where the screens are on that time the back end over there. So that's a little apps actually on the bottom left with these red buttons and green buttons. That's where we actually have to customize these little applications to copy the data and convert and rename in the way that the back end can understand. I just want to highlight is not only the back end and front end, but there's other little parts that need to be set up to make sure that the things is working. So if we want to zoom out into the higher overview, so let's come back to that previous loop. So I roughly break down into like 4 major parts of the function MRI neurofeedback setup. So most and foremost, the important the components is MRI scanner. So it's very, very important, and that's why today we're lucky we have her back. She can later on in her talk to talking about what is actually the scanner can help and how to play the roles in the neurofeedback setup. And then other equipment, including these external servers, or call them sometimes the file servers, etc. So it's connecting between the scanner and the backend to transfer the files in between. And also the backend, which is used to the real-time computing the activations for each volumes. And also this in charge of defined the region of interest, and also have to be careful of the timing. So it's very, very, you need to compute all of the information very fast. And then the front end is the one with the one presenting for the participants. So here you need to decide what type of feedback, and also what is the neurofeedback paradigm you need to consider, and what's the experiment design and other outcome measurements, etc. So for all of these questions, most of the times you actually can answer it by literature. So say literature is your best friend. So Ethan did a lot of work on the setup literature reviews and into helping to format the experiment design just from looking at other people's how they're working on it. So Ethan is definitely going to talk about it in his talk later on, so related with how the literature review can help you to build up your scenario. And this is a scanner, so this is a typical scanning sequence. I'm not going to more details about it. It's a typical structure and a functional sequence. So here we use TR equals to nearly 1 second. So we really lucky, so we are working on this 70 and then with the multi-band acceleration, so we can achieve it. So compare with about 10 years ago when we do it in Monash, the TR is about 2.5 seconds. It takes 1 TR to collect the data, another TR to calculate. So the feedback is actually 5 seconds ago. So it's not as feels real as this one. And the other equipment is pretty similar as what you set up as a functional MRI task. 3T and 70 have both experience. It feels quite similar in terms of the setup. And this part is, as I said, is connecting between the scanners and then the file, and then the backend. So it's providing the real time to pushing the DICOM files from the scanners into the backend. And this need a lot of work with the consoles to make sure that you actually can achieve this. Yeah, so they may have little variants of a different function depending on what the backend it is. The most important thing is they want to keep as fast as possible. Sometimes even just network drive will do the job. And then the next important part is the backend. So it's for the real-time computing system to analyze data and send the feedback. So we have a couple of options here. So Turbo Brain Voyager is the one we use, and we find it's quite easy to be used. It's a licensed software, but they have really good support, and then it's pretty much can handle at least a Siemens scanner very well. The front engine is the one we used before. It's actually calling like FSLs to do all of the pre-processing inside. It's good or bad of that because you take full controls of the backend, so you can customize it if you like. And another study is recently, from the Young's studies, they use FMRI, can have the similar function. And the next important thing is to decide what is the target brain regions, okay, the ROI. So there's a couple of ways you can do it. Firstly, you can come in from the prior knowledge. If you run a similar task function MRI before, and then you know what's the brain regions that related with cognitive functions, you can simply find the MRI coordinates, draw spheres, and sample the data within that regions, or you can just check the literatures. And also, if you like, you can do the customer region of interest. We call it localizer. So what we're doing is we can run a certain task right before the neurofeedback, and then we can find the person's activations of particular response to that cognitive functions and use that as the feedback regions of interest. So this is the examples of our curation task. So we get the participants to visualize some pictures with cannabis cues and the neutral cues. And then we're trying to find their activations of response to the cannabis cue, which is related with cravings. And from our previous literature, from previous work, we actually can find the cannabis users have a strong activation of ACC. There's an example of how we define, like, lock into ACC as our region of interest. And also from Ethan's review, also find ACC is a key importance of a lot of literatures. So with the overlaps of all of this, so that's why we find the initial ACC. And then we also have the custom localizers in there, so further select which brain region is that for that person's on that day, and then that will be fitting back into as used as the ROI for backend. This is examples of what is the backend is working. So as I said, timing is important. It's always a finish within 1 TR. That's our target. As you can see, the Turbo VNC is actually run pretty much pre-processing at the same time as the data is coming in. So they do the card registration, normalization, etc. And on the bottom right corners is telling you how long does it take for each volumes. So it's around 100 milliseconds. Seldomly, they're over 1,000 milliseconds, so that's what will cause 1 or 2 TRs delayed. But in general, it's doing the good job. And then the output is simply some plain text files with the activation and the percentage change in there. And then that's ready for any type of front-end software to extract the data. Yeah, the last bit part is the front-end, which is very similar. Like you can use pretty much all of the software that you would like to use for the task-based function MRI. We use Cycle Toolbox. You can either use a CyclePy. and then the previous one we use Unity together with Friend Engine. So you also need to think of what type of feedback it is. So the thermometers or the bars going up and down is one of the commonly used, and also you can think of the other ideas. For example, we have discussions about whether the feedback can be changing the brightness or actually the size of the image, is trying to use a different way to present the feedback. The next important thing is how do you design the neurofeedback paradigm, and what's your different runs, and then the up-regulation, down-regulation, or the restings. So we have this very complex design. I think maybe Amir is going to spend some time in the detail, but roughly you can see we have in this line, you have a structure imaging and followed by correction tasks. As I would say, the localizer, and then trying to find the activation for the cravings for that particular person on that day. And then the region of interest will be feeding the backend for the neurofeedback runs. And they have a resting, which is the neutral part, and also the regulation part, so we can detect the contrast. And also we have this, this we call it a VAS test, which is we asking question what persons in the scanners ask subjective questions about your craving. So you have to think of other outcomes that you want to incorporate with your design. So I would highlight the CRET neurofeedback checklist proposed by Ross, etc., in 2020. So it's a really good checklist of how you report your neurofeedback data, but also it's a really good guidelines when you start to thinking of how to design your study, and you have to set up all of the contrasts, etc. So to summarize, so I just listed quite a few of the challenges of how to do the hardware and the software setup. And really important for functional MRI, for the neurofeedback scenario is trying to produce the feedback within 1 TR, ideally. And then we can choose the proper target regions, and then also be really careful of the neurofeedback design. So all of this is the challenges, and then the solutions. Again, so literature is a better friend, and also we're going to benefit a lot from the previous projects, especially set up with the same scanners. Like we carry a lot of prior knowledge from Sampras studies in the 70s, and also you need to have a lot of dry runs and also lots of pilot scans. So for our study, I think we have more pilot scans than the real scans, in my experience. Yeah. All right, so last I'll just give, this is the updated list of the talk today, which is the same as the one on the door. You feel free to grab a printout as well. So after my talk, Sampras is going to talk about the real-time neurofeedback of his project setup, and then we'll have a question time together. And then Amir and Beck will be talking about the technical part of our setup. One is focusing on the general technical part, and Beck will be focusing on the scanner and then the console part. And then we can have a special panel discussion point, and also we prepared a little bit of quiz, so it's related with the talk. So really we cannot we don't have a scanner here to get people to fully run, but we hope through this quiz we can stimulate some discussions, and then to let people know a little bit more about how to set up into details. And after a quick break, Ethan is going to come back and talk about his literature reviews, and Elena is going to deliver her particular scenarios on depressions. And unfortunately, she's going to we're going to put a video on here because she's not here today. And at the last, we will have another very unique sessions. Ethan and Mir is going to talk about lots of practical experience and examples of how they working on and their experience and the lessons they learned of set up the back end and the front end. Okay, and then followed by the final quiz and then panel discussion. Okay, so finally, I just thank you for everyone's involved in the neurofeedback projects within Neuroscience of Addiction Mental Health Program. And thanks to Sampras, Beck, Brad, and Andrews from Uni Melbourne to support this project. Thank you very much.